Welcome back to the deep dive. So today we are opening up a chapter that I think, well, I know a lot of people, even really dedicated medical students, tend to just, skim over.
Oh, absolutely. They see the names and just panic.
Right, because the drug names are incredibly long, the life cycles are super complicated, and frankly, the organisms themselves are kind of terrifying.
They really are.
Yeah, so we are talking about the chemical warfare that we wage against parasites.
It is definitely a terrifying battlefield, but it's also, and I really mean this, one of the most intellectually satisfying ones.
Yeah.
If you actually stop to look at the underlying mechanisms, because this is a very, very different kind of war than the one we fight against bacteria.
For sure. So I was reading through the source material for this, and we are using Murray's Medical Microbiology, specifically Chapter 71. That is our absolute spine for today.
Right, sticking close to the text.
Exactly, with some, critical backup from Jowetts to help us really visualize those messy life cycles. And the metaphor that kept popping into my head, the one that I think really sets the stakes for this whole discussion is a civil war.
A civil war. I like that. That's a very apt metaphor.
Because if you think about it, right, when you treat a bacterial infection, it's kind of like sending a sniper to take out an alien target.
Because they're prokaryotes.
Right. Bacteria are prokaryotes. They are aliens compared to our cells. Their ribosomes are completely different. Their cell walls are different. Their DNA machinery is different. So you can shoot to kill the bacteria and be reasonably sure you aren't going to, hit the human cells nearby.
You have a lot of unique targets to aim at.
Exactly. But treating a parasitic infection...
The enemy looks almost exactly like the host.
Yes. Protozoa and helminths, the worms, they're eukaryotes, just like us.
They have a nucleus. They have mitochondria.
Right, they have similar organelles. So the real challenge for the pharmacologist is how do I design a poison that kills this eukaryotic invader without completely melting the eukaryotic patient?
You've hit the nail on the head. That is the core tension of this entire deep dive. It's selective toxicity.
Selective toxicity.
Right. We are operating with very, very slim margins here. The therapeutic index, meaning the gap between the dose of the drug that cures you and the dose that kills you, is often razor thin. So unlike antibiotics, where we might have this massive safety margin, with antiparasitics, we're often exploiting just tiny, tiny windows of difference.
Tiny windows. Your enzyme is shaped maybe 2% differently than my enzyme kind of window.
Precisely. Or maybe the parasite is just a little greedier for a certain nutrient. Or maybe it lacks a specific enzyme to break down a toxin that we humans can just scrub away easily. We're constantly looking for those little structural and functional wedges that we can drive a drug into.
So here is our mission for today's deep dive. We are going to navigate through Murray Chapter 71. But we aren't just going to list drugs.
Please don't. That's the worst way to study.
Right. It's the best way to fall asleep and the fastest way to fail an exam. Instead, we're going to build a structure-function mental model. We really want to give you the tools to crush those single best answer exams.
We want to move beyond just rote memorization to actual mechanism-based reasoning. I mean, if you understand how the worm moves, literally the physics of it, you understand how to paralyze it.
Exactly. And if you understand what the malaria parasite eats, you understand how to poison its food.
Exactly right.
So here is the roadmap for you. We're going to start with the protozoal battleground. So that's malaria, leishmania, giardia, the single-celled threats.
Right.
Then we'll move over to the helminthic siege, the worms, flukes, and tapeworms. That's where the strategy shifts away from chemistry and more into physics.
Yes, the macroscopic guys.
Right. And then we'll wrap up with the evolutionary arms resistance. And then, as always, we will put you to the test with a rapid-fire quiz at the end to lock it all in.
Sounds like a solid plan. Let's get right into it.
Okay, let's start with the challenge of that eukaryotic target we mentioned, the friendly fire problem. Historically, antiparasitic drugs were essentially just poisons we hoped would kill the parasite slightly faster than they killed the human, right?
That is historically very accurate. I mean, if you look at the early days of tropical medicine, we were using heavy metals, arsenic, antimony.
Which is wild.
Yeah, these are not benevolent substances. These are things you use to kill rats or, an Agatha Christie novel to take out a wealthy relative.
And we were literally injecting this into people.
We actually still do in some specific cases. And the reason this is so difficult, as we touched on earlier, is the shared biology. Both humans and parasites share nucleic acid synthesis pathways.
Hmm.
We both metabolize carbohydrates via glycolysis. We both have neuromuscular functions.
Right, so if you throw a wrench into those gears.
You risk jamming the human's gears, too.
Exactly. So how do we actually achieve differential toxicity? Murray outlines 3 main strategies, and I want to break these down for you because they apply to everything we're about to discuss today.
They're the foundation of the whole chapter.
Right. So the first strategy is preferential uptake.
Right. So imagine the parasite is like a really greedy guest at a buffet. It takes up the drug much more actively than the host cells do. It effectively concentrates the poison inside itself.
Okay.
This is how a lot of those heavy metals work, and it's actually how chloroquine works in malaria. The drug might technically be toxic to human cells too, but the parasite eats so much of it that it reaches a lethal concentration inside the invader while remaining at a completely tolerable level in the host's bloodstream.
Okay, so strategy 1 is basically the greedy guest. I like that. What's the second strategy?
The second is metabolic alteration, and this one is really clever. The drug acts as a prodrug.
Meaning it's inactive at first.
Exactly. It is technically harmless when it enters the human body. But the parasite has a specific enzyme or a specific metabolic capability that humans completely lack. And this enzyme activates the drug, turning it into a lethal toxin only inside the parasite.
So it's like a suicide pill that only the parasite knows how to swallow.
That is a perfect way to put it. We see this with drugs like metronidazole. The human body just doesn't have the machinery to pull the pin on the grenade, but the parasite does.
I love that visual. And the third strategy?
That would be target susceptibility. This relies on very subtle evolution. For example, both humans and worms have tubulin. That's the protein that makes up the skeleton of the cell, the microtubules.
Right.
But the shape of the worm's tubulin is just different enough, maybe just a few amino acids are swapped around, that a drug can bind to it and block it, while the human tubulin remains mostly unaffected.
So it's a lock and key issue. Our keyhole is shaped slightly differently, so the drug doesn't fit us, but it fits their keyhole perfectly.
Exactly.
Okay, before we dive into the specific drugs, Murray sets the stage with the global context. And I think it's really important to remember why we are actually learning this. This isn't just an academic exercise.
No, not at all. It is a matter of sheer numbers and massive human suffering.
Yeah.
As Jowitz points out in its overview, parasitic diseases rival tuberculosis and HIV in terms of global burden. We are talking about malaria alone, killing hundreds of thousands of children every year.
Yeah.
We are talking about schistosomiasis, hookworm, filariasis. These diseases absolutely thrive in conditions of poverty, poor sanitation, and in places where vaccines simply don't exist yet.
And Murray explicitly mentions this concept of mass drug administration, or MDA. This isn't just a doctor treating a single patient in a clinic. This is public health on a massive, massive scale.
Precisely. For diseases like lymphatic filariasis or onchocerciasis, which is river blindness, the strategy isn't to wait for someone to show up sick. The strategy is to literally treat the entire population of a village or an entire region, maybe once or twice a year, just to break the chain of transmission entirely. You basically flood the community with antiparasitics to wipe out the human reservoir.
Which makes understanding these drugs even more critical. I mean, if you are giving a drug to 1 million people at once, you really need to understand its toxicity profile. You can't have a 1% rate of severe side effects if you're creating a whole country.
Exactly. At that scale, the safety profile becomes just as important as the efficacy.
All right, so let's march into section 2, the antiprotozoal agents. And we're calling this the metabolic disruptors because, generally speaking, protozoa are single cells that divide really fast. So kind of like cancer or like bacteria, we try to mess up their ability to grow and replicate.
Correct. And to start, we really have to look at the oldest, clunkiest, and scariest weapons in the arsenal, the heavy metals.
Arsenicals and antimonials, I mean, these literally sound like something from the 1800s.
They essentially are. The text highlights melarsoprol and sodium stibogluconate. Let's unpack melarsoprol first. This is an arsenical. Chemically, it is trivalent arsenic.
Wait, actual arsenic, like the poison?
Literally arsenic.
Wow. Okay, so how does poisoning someone with arsenic actually cure them? What is the biochemistry there?
It all comes down to energy. Every cell needs energy, and usually that's via glycolysis. Now, there's a crucial enzyme in that pathway called pyruvate kinase. And this enzyme has these little chemical handles sticking off it called sulfhydryl groups.
Okay, sulfhydryl groups.
Right. And arsenic absolutely loves sulfhydryl groups. It binds to them incredibly
Right.
tight.
And that binding just it destroys
It physically changes the shape of the enzyme so it can't function anymore. It can completely gums up the machinery. So it essentially shuts down the parasite's entire power plant. No glycolysis means no ATP, which means cell death.
But wait, humans have pyruvate kinase too. We rely on glycolysis.
Right.
We have sulfhydryl groups.
We absolutely do. And that is exactly why Malarsapril is so incredibly dangerous.
Right.
It is highly toxic to the host. It damages neurons, it damages kidneys, bone marrow. Basically, any human cell that is highly metabolically active is going to suffer.
I read in the text that this drug is so caustic, they actually have to dissolve it in propylene glycol, which is basically antifreeze, and inject it through a special glass syringe because it melts standard plastic ones.
That is true. It is incredibly painful upon injection. It causes terrible phlebitis. But here is the key clinical pattern recognition for you for the exam and for practice. You only ever use Malarsapril for late-stage African trypanosomiasis, also known as sleeping sickness.
Let's distinguish that. Why specifically late stage?
Because African trypanosomiasis has two distinct stages. First, you have the hemolymphatic stage. That's when the parasite is just in the blood and the lymph nodes. You get a fever, you get swollen lymph nodes. But eventually, if untreated, the parasite crosses the blood-brain barrier and actually enters the central nervous system. That's the late stage. That's when you get the severe confusion, the sleep cycle disturbances, and eventually the coma.
So if I see a case vignette with a patient who has sleeping sickness and is acting confused, lethargic, maybe slipping into a coma, that's a huge red flag for CNS involvement.
Correct. And here's the catch. Most of our safer drugs cannot cross the blood-brain barrier. It's a fortress. But Malarsapril is one of the very few drugs that can cross that barrier. It is the absolute nuclear option. We use it strictly because we have no other choice to reach the parasite in the brain.
But poisoning the brain with arsenic sounds, incredibly risky.
It is extremely risky. About 10% of patients develop what's called a reactive encephalopathy, which is basically massive brain inflammation. And roughly half of those patients actually die from the treatment itself.
Wow. So you are literally making a calculation. The parasite will definitely 100% kill you. The drug only might kill you, so we have to take the drug.
That is the grim calculation. If you catch it in the early stage, before it hits the brain, we use much safer drugs like pentamidine or suramin. They don't cross the barrier well, so they are useless for the brain, but they are much, much safer for the rest of the body. Malarsapril is purely the last resort.
Okay, that is a really heavy start. Let's look at the cousin of arsenic, antimony, specifically the drug sodium stibogluconate.
It's a very similar concept. It also disrupts glycolysis by binding to those sulfhydryl groups, but the clinical target is completely different. We use antimonials for leishmania.
Let's pause there. Jawitz talks a lot about life cycles. Leishmania is the one transmitted by the sandfly, right?
Yes. Leishmania is transmitted by the bite of an infected sandfly. And when it gets into the human body, it does something very bold. It actually lets itself be eaten by our own immune cells, specifically the macrophages.
It wants to be eaten by the thing designed to kill it.
It thrives on it. Once it gets inside the macrophage, it sheds its tail as flagellum and curls up in this little tough ball called a mastigo.
Right, a mastigote meaning no whip.
Exactly. It hides inside the very cell that is supposed to destroy it. It makes its home in the phagolysosome.
So the drug sodium stibogluconate has to somehow get inside the macrophage to work?
Exactly. And that is exactly why it works. Macrophages are phagocytes. Their whole job is to eat things, so they readily eat the drug from the bloodstream. But you have to remember, antimony is highly toxic to the human heart. It causes arrhythmias, and it's toxic to the pancreas. It's not a free ride by any means, but it is very effective at concentrating inside the macrophage and killing those hiding mastigotes.
Okay, got it. So arsenicals Malarsapril are for sleeping sickness in the brain. Antimonial stibogluconate are for leishmania, hiding in the macrophage. That's a great exam anchor.
Definitely.
All right, now let's move to probably the most famous battleground of all, the malaria wars. The aminoclinolines.
This is the section where you really, really need to visualize the biology, because if you understand what the malaria parasite eats, the pharmacology makes perfect sense.
The food poisoning strategy. I absolutely love this concept. Walk us through the digestive system of a malaria parasite.
Sure. So the malaria parasite, Plasmodium, lives inside your red blood cells. And what is a red blood cell full of? Hemoglobin.
Right.
That is the parasite's primary food source. It gulps down hemoglobin into this specialized organelle called the food vacuole. It digests the protein part, the globin, to get the amino acids it needs to grow.
But there's a problem with the leftovers.
Huge problem. The center of the hemoglobin molecule is heme. And free heme is incredibly toxic. It acts almost like a detergent. It shreds lipid membranes. It produces massive amounts of free radicals. So the parasite is effectively eating a lobster where the meat is delicious, but the shell is made of pure cyanide.
So how on earth does it survive its own dinner?
Evolution. The parasite has evolved an enzyme that takes that toxic free heme and safely stacks it up. It polymerizes it into this long, inert crystal called hemozoin. Yes. If you look at a malaria parasite under a light microscope, you can actually see this dark, brownish pigment inside it. That pigment is the stack of safe, crystallized heme. It's essentially the parasite's toilet.
Enter chloroquine.
Right. Chloroquine is chemically a weak base. That is critically important. As a weak base, it easily crosses the membrane and enters the parasite's food vacuole. Now, the food vacuole is highly acidic. It's a stomach, after all.
Okay.
When the basic chloroquine hits that highly acidic environment, it gets protonated. It takes on an electrical charge.
And charged molecules can't easily cross lipid membranes to get back out.
Exactly. It becomes ion-trapped. It enters easily but cannot leave. So it begins to concentrate to massive, massive levels inside the food vacuole. And once it's trapped in there, it binds directly to the growing end of that hemozoin crystal. It caps it.
So it stops the stacking process.
It completely stops the polymerization. Suddenly, the parasite is still eating, but it has absolutely nowhere to put the toxic heme waste. The free heme builds up rapidly, generates massive oxidative stress, and the parasite effectively dies of metabolic constipation and its own toxicity. It just dissolves from the inside out.
That is such an elegant mechanism. We use their own waste product against them. But the bad news is we know chloroquine doesn't work everywhere anymore. In fact, in most places in the world, it's completely useless against P. falciparum.
Sadly, yes. Resistance is incredibly widespread. And this brings us to a key exam point that you have to know, the mechanism of that resistance. It is driven by a gene called PFCRT, the Plasmodium falciparum chloroquine resistance transporter.
I see the word transporter, so it's a pump.
It is a mutated protein sitting right on the membrane of that food vacuole. It acts as a customized efflux pump. It literally grabs the charged chloroquine and pumps it right back out of the vacuole into the parasite's cytoplasm before it has a chance to cap the crystals.
So the drug gets into the stomach, but the parasite just bails it out immediately.
Exactly. And because the drug can't accumulate to toxic levels, the parasite can just keep stacking its heme safely and goes right on living.
Man. So if chloroquine fails, or if we have a really severe case of malaria, we have to bring in the heavy artillery, quinine and quinidine.
The cinchona alkaloids, yes. These are historically derived from tree bark. They are extremely potent, and they actually work very similarly to chloroquine, but they target slightly different steps in the crystallization process, so they can still work against many of those resistant strains. But they are very messy drugs.
Messy? How?
The side effect profile, we call it ingenism. Patients get ringing in the ears. So that is severe headache, nausea, blurred vision, and much more dangerously, quinidine is actually a class 1A antiarrhythmic drug.
Oh, right.
It prolongs the QT interval on an ECG. You can literally stop the patient's heart if you aren't infusing it carefully and monitoring them.
That is a classic board exam trap. A patient gets treated for severe malaria and then suddenly passes out or has a fatal arrhythmia. You always check the QT interval.
Precisely. Now, sticking with the quinolines, there is one specific drug in this class that acts completely differently, and you absolutely must know it for the exams.
Primaquine.
The root killer. Yes. To fully understand primaquine, you have to look back at the life cycle maps in Jayowitz. Most of our malaria drugs, chloroquine, quinine, artemisinin, they strictly kill the parasite when it's in the red blood cells. That's the erythrocytic stage. That's what actually causes the clinical symptoms.
Yeah.
The fever, the chills, the anemia.
But two specific species of malaria, Plasmodium vivax and Plasmodium ovale, they have a superpower.
They can sleep. When they first enter the human body from the mosquito, some of them go straight to the liver and just, they don't divide. They become hypnozoites.
Yeah.
Hypno meaning sleep.
Right.
So you can clear the entire blood infection with chloroquine. The patient's fever breaks, they feel totally fine, and they go home. But 3 months later, or even a year later, those hypnozoites wake up, leave the liver, invade the blood all over again, and the patient relapses.
Without ever getting bitten by another mosquito.
Without a new bite. It's the exact same original infection, just waking up from its nap.
So primaquine is our weapon against those sleeper cells.
Correct. It is currently the only available, widely used agent that is effective against the litter hypnozoites. This strategy is called a radical cure. So if your exam question features a traveler returning from the tropics, maybe a soldier coming back from deployment or a backpacker from Southeast Asia who gets treated for malaria, gets better, but then keeps getting sick again months later, the answer is they were not treated with primaquine for a radical cure.
And how does primaquine actually kill them in the liver?
The exact mechanism is still a bit debated, but we think it generates highly reactive oxygen species that interfere with the parasite's electron transport chain in its mitochondria. But here is the absolutely critical safety warning for the wards. Because it causes systemic oxidative stress, you cannot just hand this out to everyone.
Ah, this is the G6PD connection.
Exactly. Glucose-6-phosphate dehydrogenase deficiency is a very common genetic condition, especially in areas where malaria is endemic. These patients' red blood cells are extremely sensitive to oxidative stress because they can't regenerate glutathione.
Okay.
If you give primaquine to a G6PD deficient patient, you will cause a massive, sometimes fatal, hemolytic crisis. Their red blood cells will literally burst under the oxidative strain.
So, rule number one before even thinking about prescribing primaquine, check the patient's G6PD status.
Always. Every single time.
All right, moving on to the next class, the antiphalates. Now, we use these for bacteria too, right? Like sulfonamides and trimethoprim. Why do they work on eukaryotic parasites?
The underlying principle is identical to how they work in bacteria. It's all about DNA synthesis. To make DNA, any cell needs folate, which is vitamin B9. Now, humans get our folate from our diet. We eat spinach, we eat fortified cereal. We have a dedicated transport system to pull dietary folate right into our cell.
But parasites can't do that.
Most parasites, just like bacteria, cannot import pre-made folate. They are totally impermeable to it. They have to build it themselves from scratch using a chemical precursor called PEVA. They are essentially miniature folate factories.
So we just sabotage the factory.
Exactly. We use a synergistic, two-pronged attack. The drug sulfadoxine chemically mimics PEVA. It acts as a competitive inhibitor and blocks the very first enzyme in the assembly line, which is dihydroapatite synthase.
Got it. Step one, blocked.
Then we add a second drug, pyrimethamine, which blocks the second major step, specifically the enzyme dihydrofolate reductase.
So 1 + 1 = 3 here.
Pure synergy. By blocking two sequential steps in the same pathway, you shut down DNA synthesis completely. This specific combination, known as Fansidar, used to be a massive tool for treating malaria. Unfortunately, point mutations have caused resistance to weaken it significantly, but this antifolate mechanism is still the absolute go-to for treating another major parasite, Toxoplasma gondii.
Right, the one associated with cat feces in pregnant women. Okay, this next section is honestly my absolute favorite because it feels like a bizarre plot twist in a sci-fi movie. Protein synthesis inhibitors. We are talking about classic antibiotics here. Doxycycline, clindamycin, drugs that specifically target the bacterial 30S or 50S ribosome.
Right.
But as we established in the very beginning, parasites are eukaryotes. They have 40S and 60S ribosomes, just like human cells. So why on earth would giving doxycycline kill a malaria parasite?
This is one of the most fascinating stories in all of evolutionary biology. It turns out the malaria parasite has a tiny hidden organelle inside it called the apicoplast.
The apicoplast.
Yes, it's a vestigial organelle. Millions and millions of years ago, the evolutionary ancestor of the malaria parasite actually swallowed an algae, a plant-like organism, and that algae had previously swallowed a bacteria. Over eons of evolution, they merged. But that tiny apicoplast still retains its own circular DNA and its own ribosomes, which are functionally bacterial in origin.
So the malaria parasite basically has a tiny ancient bacteria living inside it that helps it function.
Exactly. The parasite absolutely needs the apicoplast to manufacture essential fatty acids. So when you give a patient doxycycline, you aren't targeting the parasite's main nucleus or its main eukaryotic ribosomes. You are targeting this ancient, swallowed bacterial organelle. You shut down the apicoplast, it can't make fatty acids, and the parasite dies.
That is just incredible. It's like finding out the Death Star has a tiny thermal exhaust port that runs on Windows 95.
Ah, yes, and we can exploit it beautifully. But there is an even cooler example of antibiotics killing parasites in this chapter, river blindness.
Oh, the bank shot strategy.
Yes. So the nematode worm Onchocerca volvulus is what causes river blindness. But it turns out the worm itself is actually infected. It has a bacterial tenant living deep inside its own gut and tissues, a bacteria called Wolbachia.
Bacteria living inside a worm, which is living inside a human. It's a literal Russian nesting doll of infection.
And they are obligate endosymbionts. They are life partners. The worm absolutely relies on the Wolbachia bacteria for essential nutrients and actually for normal embryogenesis. If you take away the bacteria, the worm suffers terribly.
So we don't shoot the worm, we shoot the bacteria inside the worm.
Exactly. You give the human patient doxycycline. The doxycycline kills the Wolbachia bacteria. Without its bacterial partner, the adult female worms become sterile, stop producing larvae, and eventually die. It's a brilliant strategy because you're targeting a prokaryotic target, the bacteria, to kill a eukaryotic enemy, the worm. It's vastly safer for the human patient because our cells don't care about doxycycline.
Science fiction level stuff. I love it. Okay, let's quickly hit the diamidines and nitroimidazoles before we shift over to the worms.
Sure. Pentamidine is the key diamidine to know. Its exact mechanism is honestly a bit messy. It binds DNA, it interferes with polyamine synthesis, but the key clinical distinction you need to make goes right back to sleeping sickness.
Right. We said melarsoprol is for the brain, the late stage.
Right. Pentamidine is for the early stage, or the hemolymphatic stage. That's before the parasite has crossed the blood-brain barrier. Pentamidine is a large, highly charged molecule that does not cross the BBB well at all. So it's much less toxic to the patient than melarsoprol, but it's completely useless if the infection is already established in the central nervous system.
That makes perfect sense. And what about metronidazole?
The absolute classic, the mnemonic every student needs is, get on the metro.
Ah, right. G for Giardia, E for Entamoeba, T for Trichomonas.
Correct. And what do those three have in common? They are all anaerobic protozoa. Metronidazole is another classic prodrug. It is completely harmless, floating around in your blood until it enters an anaerobic cell. These specific parasites have a unique electron transport protein called ferredoxin, and this ferredoxin system mistakenly donates an electron to the metronidazole molecule.
And that reduction activates the drug.
Yes. It turns the inert drug into a highly reactive, chaotic, free radical. It's literally like tossing a live grenade into a very small room. It just violently shreds the parasite's DNA. And since human cells are aerobic and lack that specific ferredoxin electron donor system, we don't activate the drug, so our DNA is mostly spared.
Get T on the metro! That's definitely a keeper. Now, to finish out the protozoa, we have to talk about the current absolute gold standard for malaria treatment, the sesquiterpenes, better known as the artemisinins.
This is an amazing story, too. It's derived from the sweet wormwood plant, Artemisia annua, which has actually been used in traditional Chinese medicine for centuries to treat fevers. The modern drugs are artesunate and artemether. These are incredibly powerful, exceptionally fast-acting drugs.
How do they work? Because it involves heme again, right?
Yes, it targets the food vacuole again. The drug molecule has a very unique structural feature called an endoperoxide bridge. Think of it as a highly chemically unstable fuse. When the drug enters the parasite's food vacuole and encounters all that iron in the heme, the iron acts as a catalyst and reacts with that bridge. It causes it to cleave open violently.
Another chemical explosion.
Exactly. It releases a massive burst of free radicals that instantly damage the parasite's proteins and lipid membranes. It kills them incredibly fast. I mean, it reduces the parasite load in the blood by orders of magnitude within just hours.
But there is a golden rule with artemisinins, right? You never ever use them alone.
Never. Monotherapy with artemisinins rapidly leads to resistance. We always use ACT. Artemisinin-based combination therapy.
So you pair it up.
Right. You combine the fast-acting artemisinin, which clears the massive bulk of the infection almost immediately, with a long-acting partner drug like lumefantrine or mefloquine. The partner drug hangs around in the blood for weeks to mop up any lingering survivors. You protect the artemisinin from developing resistance.
Okay, let's take a deep breath. We have poisoned their metabolism. We have sabotaged their DNA factories. We have even killed their tiny bacterial pets.
We've been busy.
Very. But now we are shifting gears. Section 3, the helminthic siege.
The macroscopic guys, the worms, roundworms, tapeworms, flukes.
The entire strategy has to shift here, doesn't it? Because protozoa are tiny, rapidly dividing single cells. But worms are. They're actual animals. They have muscles, they have a nervous system, they have an outer skin called a tegument, they have a gut. They aren't dividing rapidly in the host, they're just getting bigger.
Exactly. An adult tapeworm sitting in your gut is not replicating its DNA like a bacteria in a petri dish. It is just hanging on, absorbing nutrients, growing and laying thousands of eggs. So targeting DNA synthesis is often completely useless. We have to target their physical ability to stay inside us.
So we attack their physiology.
Right. We either paralyze them by hitting their neuromuscular junctions, we starve them by breaking their cellular cytoskeleton, or we literally burn their skin off to expose them to the immune system.
Wow. Let's start with the starvation tactic, the skeleton breakers. This is the benzimidazole class. Albendazole and mebendazole.
These are the absolute workhorses of antihelminthic therapy. Their specific molecular target is beta-tubulin.
Okay, tubulin is the protein that polymerizes to make microtubules, and microtubules are basically the cell's internal highway system, right?
Correct. And worms are exquisitely dependent on microtubules for transport within their intestinal cells. They need these microtubule highways to move vesicles to the surface to uptake glucose from the host's gut. If you block tubulin polymerization, you physically block glucose uptake.
So the worm just starves to death.
Yes, it slowly depletes its internal glycogen stores. Its energy runs out and it dies. It's a very slow death. Unlike the paralysis drugs, it takes days for them to fully die and be passed.
But again, going back to the friendly fire problem, humans have tubulin. Our cells use microtubules for cell division and transport. Why don't my cells starve when I take albendazole?
Selective toxicity is that third strategy we talked about. The drug has a much, much higher affinity for helminthic beta-tubulin than it does for mammalian beta-tubulin.
Yeah.
It binds to the worm's protein something like 300 to 400 times stronger than it binds to yours.
Okay, that makes sense. Now here is a crucial clinical pearl regarding albendazole versus mebendazole. They sound almost identical. Are they just totally interchangeable?
Not entirely, no. It all comes down to their pharmacokinetics, specifically their absorption. Mebendazole is very poorly absorbed by the human GI tract. It mostly just stays in the gut lumen.
And sometimes that's what you want, right?
Exactly. That is exactly what you want if you are treating a whipworm or a pinworm that is living right there in the intestinal lumen. You want the poison concentrated in the tube, not circulating in the patient's blood.
But albendazole...
Albendazole is absorbed much better systemically, especially if you tell the patient to take it with a high-fat meal. It gets absorbed into the bloodstream and distributes into the tissues.
Why does that matter clinically?
Because sometimes the worms aren't politely staying in the gut. Think about neurocysticercosis.
Oh, that's when the pork tapeworm larva forms cysts in the brain.
Right, or echinococcus, forming massive hydatid cysts in the liver. If the parasite is in the brain or the liver, mebendazole won't reach it. You absolutely need a drug like albendazole that can leave the gut, enter the blood, and penetrate those deep tissues.
So a good rule of thumb for the exams, mebendazole for the tube, meaning the gut, and albendazole for the tissue, meaning cysts and abscesses.
That's a very solid rule of thumb.
All right, let's move to the paralyzers. This is where the physics of it comes in. We really need to distinguish between two completely different types of paralysis, spastic and flaccid.
This is a classic high-yield exam differentiator. Do you make the worm rigid and stiff, or do you make it limp and floppy?
Let's look at parental pomoda first.
Parental is a depolarizing neuromuscular blocker. It acts essentially like acetylcholine. It strongly stimulates the worm's nicotinic receptors at the neuromuscular junction and forces them to stay open.
So the muscles fire, contract, and just can't relax.
Exactly. That is spastic paralysis.
Yeah.
The worm physically seizes up, it gets stiff, it curls into a tight little ball, and because it is completely seized up, it loses its muscular grip on the intestinal wall. Then, normal human peristalsis, the natural movement of your gut, just flushes the rigid worm right out in the feces.
Nice. Then we have the exact opposite approach, ivermectin.
Ivermectin is a truly fascinating drug. I mean, it won the Nobel Prize for its discoverers because it revolutionized tropical medicine. Its target is glutamate-gated chloride channels. These specific channels are unique to invertebrates. Mammals simply don't have them in our peripheral nervous system.
And what physically happens when you force a chloride channel open?
Well, chloride ions carry a negative charge, so negative ions rush into the nerve cell. This rapidly hyperpolarizes the cell. It makes the internal voltage so negative that it becomes impossible for the nerve to fire an action potential.
So the electrical signal just completely stops. The muscle goes limp.
Yes. Flaccid paralysis. The worm becomes completely flaccid and can't move at all. We use this heavily in onchocerciasis, or river blindness. It paralyzes the microfilaria, which are the tiny larval worms migrating right under the patient's skin and into their eyes. By paralyzing them, It stops the intense itching and halts the progression of the blindness.
Wow. Now, there's a third neuromuscular drug mentioned, diflucarbamazine, or DEC. This one seems a bit weirder.
DEC is our primary weapon for lymphatic filariasis, which causes elephantiasis, and for the African eyeworm, loa loa. It does seem to stimulate cholinergic receptors to cause some immobilization, but its most important effect is actually immunological. It alters the surface membrane of the worm.
It effectively unmasks the worm to our immune system.
Exactly. It acts like a spotlight. It exposes surface antigens that the worm was actively trying to hide. Suddenly, the host's immune system sees the worm clearly and mounts a massive attack.
And that immune attack leads to a very specific, dangerous clinical reaction you need to know, the Mazzotti reaction.
This is crucial for anyone practicing tropical medicine. Because DEC kills the microfilaria so rapidly and exposes all these foreign antigens to the immune system at once, you can trigger a massive systemic inflammatory cascade. The patient develops a high fever, severe rash, tremendously swollen lymph nodes, tachycardia, hypotension. It looks exactly like anaphylactic shock.
But it's not an allergic reaction to the drug itself, right? It's the body overreacting to millions of suddenly dying worms.
Exactly. It's a therapeutic shock. You have to anticipate it and often manage it by co-administering systemic corticosteroids and antihistamines just to calm the immune system down while the drug does its work.
Good to know. Okay, moving to mechanism 3, the tegument busters. Specifically, praziquantel. This is the absolute go-to for trematodes, which are flukes like schistosoma, and cestodes, the tapeworms.
Schistosomes are absolute masters of disguise. They actually coat their outer skin, their tegument, in host proteins. They cover themselves in your blood antigens, so the immune system just thinks they are a normal part of the blood vessel wall.
Yeah.
Praziquantel violently ruins that disguise.
How does it do that?
It acts as a severe calcium agonist. It forces open calcium channels and causes a massive, uncontrolled influx of calcium straight into the worm's tissues.
And calcium usually triggers muscle contraction.
It does. So the immediate effect is intense tetanic muscle contraction. But secondary to that, and much more importantly, this massive calcium storm physically damages the tegument. The textbook describes it as vacuolization. The worm's skin literally forms bubbles, blisters, and eventually bursts open.
That sounds absolutely horrific.
For the worm, it is. But therapeutically, it's brilliant. It violently exposes all the deep hidden antigens underneath. It blows the spy's cover, so to speak. Once that skin is blistered and damaged, the host antibodies and white blood cells rush in, latch onto the exposed tissue, and physically destroy the worm.
So praziquantel essentially skins the worm alive and just lets the immune system finish the job. Brutal, but effective.
Very effective. It's the cornerstone of schistosomiasis control globally.
Finally, mechanism 4 for the worms. The uncouplers. Specifically, niclosamide. This feels like a total throwback to first-year biochemistry class.
It really does. Niclosamide acts directly on the mitochondria of intestinal tapeworms. It uncouples oxidative phosphorylation.
Let's try to visualize that for a second. The mitochondria is basically like a hydroelectric dam, right? Protons build up on one side of the dam, and when they flow through the turbine, which is ATP synthase, they generate the energy we call ATP.
That's a great analogy. Correct. Niclosamide essentially drills a massive hole right through the wall of the dam. So the protons just flow uselessly through the hole, bypassing the turbine entirely. The whole process continues, the water flows, oxygen is consumed, but absolutely no energy is generated. The worm is burning its fuel, but making zero ATP.
It just runs completely out of gas.
Precisely. And the beauty of it is niclosamide is a very large molecule that is generally not absorbed by the human gut. It stays entirely in the intestinal lumen where the tapeworm is attached, so our own mitochondria and our tissues are completely safe.
Simple, elegant, and effective. Okay, so we have poisoned them, starved them, paralyzed them, and skinned them alive. But, as always in microbiology, the enemy is highly adaptable. Let's hit section 4. Mechanisms of resistance.
The evolutionary arms race. This is really high yield for exams because treatment failure is a very common question stem. You prescribed the technically correct drug, but the patient's fever didn't break. Why?
Let's focus on malaria first, because that's the big one. We already mentioned chloroquine and the PFCRT gene.
Right. Just to reiterate, that's the specific efflux pump located on the membrane of the food vacuole. It actively pumps the basic drug out of the acidic stomach so it never gets a chance to cap the toxic heme crystals.
Got it. But what about multi-drug resistance? Because that's a different mechanism, right?
Yes, that involves a totally different gene, PFMDR1. This is distinct from PFCRT. The PFMDR1 gene encodes what's called a P-glycoprotein homologue. Think of it as a heavy-duty bilge pump on the outer membrane of the parasite, or its main organelles. It non-specifically pumps a whole variety of bulky drugs like mefloquine, quinine, and even some artemisinins completely out of the parasite.
So PFCRT is a very specific pump protecting just the food vacuole, but PFMDR1 is a broad-spectrum pump protecting the whole cell.
Roughly speaking, yes. If you see increased copy numbers of the PFMDR1 gene on a lab report, it strongly correlates with multi-drug treatment failure.
Okay, and then there's the really big worry right now in global health. Artemisinin resistance. You mentioned earlier, this is the scariest one.
It truly is. It is primarily emerging in the greater Mekong subregion, so that's places like Cambodia, Laos, Vietnam, Thailand, and it is genetically linked to specific point mutations in what's called a K13 propeller protein.
K13. Does K13 act like another pump to spit the drug out?
No, it's actually much more insidious than a pump. K13 mutations seem to allow the parasite to dramatically alter its own developmental cycle. When the short-lived artemisinin drug is present in the blood, the ring stage parasites enter a bizarre state of deep dormancy or quiescence. They essentially just hold their breath and stop metabolizing.
Wait, they just wait for the drug to leave?
Exactly. Artemisinins are incredibly fast-acting, but they have a very short half-life. They are cleared from the body in just a few hours. So the mutant parasite just stops growing, waits safely in a dormant state for the drug to clear, and then wakes right back up a day later and continues destroying red blood cells. It's a temporal survival mechanism, not a physical blockade.
That is terrifying, honestly, because artemisinins are really our best line of defense against severe falciparum malaria.
Exactly. That is why the WHO is so hyper-focused on containment and surveillance in that specific region of Southeast Asia.
Okay. We have built the mental model. We have covered the structures, the functions, and the mechanisms of evasion. Now it's time to consolidate. It is time for the rapid-fire SBA quiz.
Let's do it. I will pose the clinical scenario. I want you to try to visualize the patient in front of you. Then I'll ask the specific question. Pause the audio, think it through, and try to answer before we give it away.
All right, listener, try to beat the expert. Here we go.
Question 1. A 24-year-old aid worker returns from Lake Malawi. She admits to swimming in the freshwater lake, despite warnings about parasites. 3 weeks later, she develops gross hematuria blood in her urine. Microscopy of the urine shows large eggs with a distinct terminal spine. You correctly diagnose schistosoma hematobium. You prescribe a drug that works by dramatically increasing calcium permeability, causing tetanic contraction, and physically damaging the worm's tegument. What is the drug?
Swimming in freshwater plus blood in urine equals schisto. What's the tegument buster?
The answer is praziquantel. Remember, praziquantel is the calcium agonist. It blisters and skins the fluke. It is the absolute drug of choice for all forms of schistosomiasis.
Nice. Next one.
Question 2. A female soldier returns from a long deployment in Afghanistan. She develops a classic pattern of fever and chills, peaking every 48 hours. A thick and thin blood smear confirms a Plasmodium vivax infection. She's treated effectively with chloroquine, and her fever resolves completely. However, the infectious disease physician adds a second drug to her regimen specifically to prevent future relapse from liver stages. What is the primary mechanism of action of this second drug?
So we need the root killer for the sleeper cells. The drug is primacaine, but what does it actually do?
The correct answer to the mechanism question is generation of highly reactive oxygen species and interference with mitochondrial electron transport. Remember, primacaine kills those dormant hypnozoites in the liver. Chloroquine is excellent, but it only kills the active blood stage.
And always check for G6PD before giving it. Question 3.
Question 3. A 6-year-old child living in a rural agricultural community presents with a painfully distended abdomen and eventually passes a large, intact roundworm in their stool. You wish to treat the child and their family with a drug that specifically binds to beta-tubulin, preventing microtubule polymerization and effectively starving the worm of glucose over several days. Which agent acts via this exact mechanism?
The skeleton breaker that causes starvation.
The answer is albendazole, or its cousin mebendazoles. The benzimidazole class targets tubulin. No tubulin equals no vesicle transport, which equals starvation.
Okay, question 4.
A patient diagnosed with a river blindness caused by onchocerca volvulus is treated with a systemic drug that causes flaccid paralysis of the microfilaria by opening glutamate-gated chloride channels. Which drug is this?
Flaccid means limp or floppy. Chloride channels hyperpolarize.
The answer is ivermectin. It's crucial to remember the difference. Pyrantel causes spastic or rigid paralysis via cholinergic receptors. Ivermectin causes flaccid or limp paralysis via those unique chloride channels.
Perfect. Last one. High stakes.
Question 5. A patient is brought to the emergency department with severe confusion, profound lethargy, and inverted sleep disturbances. He recently returned from a month-long safari in East Africa. A lumbar puncture confirms the presence of Trypanosoma brucei directly in his cerebrospinal fluid. You desperately need a drug that inhibits pyruvate kinase, but, most importantly, can cross the blood-brain barrier. It is notoriously highly toxic. What is the agent?
The absolute nuclear option for the brain.
The answer is melarsoprol. It's an arsenical compound. It crosses the BBB to treat late-stage CNS sleeping sickness. Pentamidine is for early-stage blood infection only and does not cross well.
How did you do? If you missed any of those, make sure to go back to the relevant sections in Murray Chapter 71. These core mechanisms, spastic versus flaccid, blood versus liver, brain versus body, these are the absolute bread and butter of parasitology board exams.
They really are. And they love asking about the toxicity profiles too, so keep melarsoprol's encephalopathy and primakine's G6PD hemolysis front and center in your mind.
All right, let's wrap this up. We have covered a massive amount of ground today.
We really has. We saw that protozoa are incredibly fast multipliers, so our main strategy is to hit their metabolism, disrupt their folate synthesis, poison their heme processing, shut down their glycolysis. They act a bit like cancer cells. We target their rapid growth.
Right, and helminths are slow, complex, muscular beasts, so we don't bother with their DNA. We hit their nerves to paralyze them, we hit their skeletons to starve them, or we destroy their skin to unmask them.
Exactly. It's all about matching the weapon to the physiology of the specific enemy.
Now, before we sign off, I want to leave you with a somewhat provocative thought that came to me while I was reading about doxycycline and river blindness.
Ah, the Wolbachia connection.
Right. Murray mentions that one of our single best, safest strategies for killing a massive parasitic worm is actually an antibacterial drug. It makes me wonder, as we learn more and more about the microbiome of the parasite, these endosymbiotic bacteria living inside the worms or inside the protozoa, could the future of parasitology actually just be bacteriology?
That is a truly fascinating frontier in research right now. We are actively moving from simply poisoning the host and just hoping the parasite dies slightly faster to surgically dismantling the parasite's own internal life support systems. I mean, if a deadly parasite completely relies on a benign bacteria to survive, simply killing that bacteria is the ultimate precision strike. It completely redefines what an antiparasitic actually is.
It really does. It's a civil war where, instead of bombing the battlefield, we just bribe the enemy's cook to stop feeding them.
A perfect analogy to end on.
Well, that is it for this deep dive on antiparasitic agents. Be sure to go read Murray Chapter 71. Look closely at those specific gene names, PFCRT, PFMDR1, K13. Lock them in your memory.
And always remember, selective toxicity is the name of the game.
See you next time.